Greetings. Welcome to the Evidence Reasons Academy. This is June 24th. Katie and I are going to be exploring peer-reviewed literature. We're going to do a little bit of a journal club and then start to use basic local alignments, the acronym is BLAST. And so I'm here trying to teach her kind of bioinformatic tools. And I'd say UniProt is the number one tool for the stuff we're doing. Next would be BLAST. Katie is a student of biology, a very promising one, and I'm privileged to have a chance to be to work with her on a variety of things. And also, this is a chance to educate my colleagues on the internet who are interested, the subscribers and visitors to my channel, about the sort of stuff that I do as far as biology. I know I'll have the chance to torture my audience on many other things, like in biophysics and physics and quantum mechanics and statistical mechanics. But today we'll go easy on you guys. We're going to do kind of basic bioinformatics. So this is, I'm not trying to put you on the spot, but I think when I ask questions, I could see, I could compare my understanding versus yours, Katie. Can you tell me what a nuclear localization signal does?
Yeah, a nuclear localization signal helps a protein get either back into the nucleus or leave the nucleus to get into the cytoplasm.
Very close. Technically, nuclear localization puts it in the nucleus. Mm-hmm. Nuclear export.
Yeah.
Export is what you had the right idea. Very good. So what we're going to do, this is going to be an interesting question. And since my channel is for creationists and ID proponents may or may not be old Earth creationists. Many are young Earth, some are old Earth creationists, some still believe in common descent. We're a big tent. And even though we would disagree on the chronology of history of the universe, we have broad agreement on certain things within intelligent design. And that's actually been very helpful because some of the best people in the biological sciences are old Earth creationists, or they believe in some form of common descent, like Michael Behe. But they're outstanding researchers, and we do have the common ground about criticizing Darwinian evolution. So there is, just for the audience's sake, there is an outstanding problem that I had mentioned to Dr. Daniel Stern Cardinal, and he seemed curious. I said, look, when you change the size and shape of parts in a car, especially like if it's a central piece, you're going to have to change, you're definitely going to have to change the size and shape of stuff that connects to it. So one can't in isolation just say this evolved without accounting for all the coordinated changes that have to happen elsewhere. And that actually makes the probability argument against natural evolution that much more pointed, that much more difficult. Does that sort of make sense, Katie?
Yeah.
Yeah, and I'll tell you, the more that we explore, you're going to see more and more complexes where there are more and more connections, and there could be hundreds of connections in a particular pathway. You can't just flip one without flipping a whole bunch of others. And nuclear localization. So I'm going to give just a wild guess that there are at least 200 proteins that have to be in the nucleus for the nucleus to function, at least 200 in the eukaryotic. And then maybe another 100, probably another 100 in the the membrane that surrounds the nucleus. And I think those numbers are very conservative, by the way. So we have to have all these localization signals placed on all of those, practically all those proteins. There are a few exceptions. And if you change the importin, you're going to have to change all 400
Mm-hmm.
or 300 or whatever number. How is that? How is the simultaneous evolution going to happen? And why should the Darwinian process favor simultaneous evolution when there's a saying that, have you ever heard the saying, if it ain't broke, don't fix it?
Yeah.
Okay, so if they're going to reformat, make a major improvement to the cell so it can go from unicellular to multicellular, and to be, something as complex as a mammal, there could be a lot of simultaneous changes that have to be made. It's like, again, the car analogy or anything of complexity.
Mm-hmm.
So the reason that is kind of a primer on where we're going. I want to show that this is actually true, that various species have different localization signals. And even though this is experimentally difficult, if we can show the important complex has to be radically changed between species, that means it's going to be very, very hard to evolve one creature into another. Does that sort of make sense?
Yeah, that makes sense.
It's an argument. We can still say, look, for the sake of argument, we'll accept common descent. And for the record, I believe the universe is young, the earth is young, life is young, the universe is young. There's some problems in that model from a scientific standpoint, but that's something I've come to believe. And people know that I used to believe in evolution, and that went out the door when I just got a little exposure to the origin of life problem. And I was an old Earth creationist most of my life, and then became a young Earth creationist. So that's just setting the record straight. But for the sake of argument, we'll just say let's just suppose that there is common descent. How hard would it be to transform something like a single-celled organism, like a eukaryotic organism, like a aspergillus, or whatever evolutionists think is the closest relative to a human from the single-celled eukaryotes. What if we found that their nuclear localization signals are radically different? That's where we're headed. Now, I had the privilege of meeting Katie backstage, and I can tell, when she turns on the camera, I can tell if she's frowning or smiling. But when her camera is off, I can't tell that. So you got to tell me, Katie, does that sound even remotely interesting?
Oh yeah, it definitely does.
Okay, then, all right, we're on then. Okay, so I'm going to have to give you some hard lessons here. And we already talked about this. So let me download, let me get my screen share going. And can you see my screen?
Yeah, I can see it.
Okay, I'm going to download a PDF. I'll just put it in the junk folder for now. I'm going to grab Aspergillus topo. Aspergillus topo. Where did I put this?
It's right at the top. Do you see it? In the Chrome?
Identification of a single nuclear localization signal in the C-terminal domain of Aspergillus. Okay, so we're going to read parts of this. Hey, you have really good eyes. I couldn't find. Thank you for finding that in all of that. Do you watch sports by any chance?
Not really.
Did you play any in high school?
I played softball.
You do look a little athletic.
Yeah, I mean, I like to run sometimes, but I'm not super athletic.
Well, I was a little embarrassed because you saw my um, sort of crazy stuff that I watch. Like that's there's this anime here that was it was like a 5 year series and it finally ended in a wedding.
Oh.
So the girl had this was in love with the captain of her ship, and I have that. You'll see it. I put it actually on my channel. And then there's Sophie Cunningham. She's a basketball player. I just
Mhm.
had to explain that. She's on the WNBA.
Mhm.
She plays for the Fever. So anyway, I normally don't watch women's basketball, but Caitlin Clark is her teammate, and
Mhm.
everyone started watching the WNBA because of Caitlin Clark.
Yeah.
Anyway, so and I do have a twisted sense of humor. This was this was okay. I just have to go here. Sorry.
It's okay.
So let me just show you the picture. So as you know, there was a bomb dropped. President Trump ordered the B-2 bombers.
Oh.
I'm sorry. Okay. So I better watch what I let you see on my screen because then you'll know all the crazy stuff I'm into. So anyway, can you see this?
Yeah, I can see it.
Um, all right. I'm going to just have you read several sentences. Can you see it clear enough to read here?
Yeah.
Yeah. Okay,
You
just
want me
read till the about the bottom.
Okay. DNA topoisomerase 2 is a major nuclear protein that plays an important role in DNA metabolism. We have isolated the gene for topo2 from the filamentous fungus Aspergillus terris. Hopefully, I pronounced that right. The deduced amino acid sequence revealed that topo2 consists of 1,587 amino acids and has a calculated molecular weight of 180. I'm not sure what that is.
Kilodaltons.
Okay. The
Okay,
pro-
no, stop.
hmm?
This is fine. Um, we only have a few words we didn't understand, right?
Yeah.
Okay, good.
Okay.
Go on.
The protein expressed in Escherichia coli has an estimated molecular weight of 185 kilodaltons. Expression of topo2 polypeptides tagged with yellow fluorescent protein in building yeast suggests that the C-terminal region of the topo2 is essential for transport of the fusion protein into the nucleus. The nuclear localization signal sequence of topo2 is a non-classical bipartite type. Okay, I don't know what that means.
Mm-hmm. It's fine.
Containing two interdependent positively charged clusters separated by 15 amino acids. Alanine scanning mutagenesis and deletion analysis showed further that a stretch of 23 amino acid residues is necessary for nuclear import. In addition, we confirmed using co-immunoprecipitation and two hybrid analysis that this non-classical nuclear localization signal interacts with important alpha in building yeast. These results suggest that the fungal topo2 nuclear localization signal is a functional in yeast cells. Do you want me to keep going?
Um, actually, No, that's fine. This was okay. So, how painful was that on a scale of 1 to 10 of anything you've done in school?
Not too bad.
Okay, good.
There were only a couple, like towards the bottom, what they were talking about towards the bottom wasn't very familiar with me, but like the first half of the abstract made sense for the most part.
This is actually one of the best papers I've read.
Mm-hmm.
They go through a lot of, I mean, the writing style is very clear.
Mm-hmm.
So, no, then I'm really glad because this is a blessing to have because, I mean, just starting out. So, well, why don't we go through more? So we'll go through the introduction.
Okay. You want me to
Can you
read?
see it okay? Mm-hmm.
Yeah, I can see it.
Sure, go ahead and read.
Okay. DNA topoisomerases are ubiquitous enzymes that are found in all prokaryotic and eukaryotic organisms. Because of the intertwined nature and the duplex DNA, topological problems arise during DNA metabolism. DNA topoisomerases are designed for the resolution of different DNA topological forms, and they participate in many vital cellular functions, including DNA replication, transcription, recombination, chromosome condensation and segregation, and DNA repair. These enzymes have been classified into two types, 1 and 2, according to their catalytic mechanism. DNA topoisomerase 2 open and close gaps in double-stranded DNA for the passage of other double-stranded DNA segments. Members of this family include DNA gyrase and bacterial topo 2.
Good. By the way, I only got this paper last night, so it's really good. I mean, I've been hunting for papers for, for us to work on. Do you remember what topoisomerase does?
It prevents like supercoiling from happening during I guess all the processes that it described in the introduction, like transcription, translation, recombination, condensation, chromosome condensation. But it helps like supercoiling of the DNA so it doesn't get too
That
tight.
is fabulous.
Yeah.
So let me see if I have this. By the way, you listed off all that stuff with recombination and segregation and condensation by memory. I'm impressed.
Thanks.
Okay, so I'm going to show you something. I'd encourage you not to visit this place.
Okay.
You'll get to hear all the people that hate me on the internet. There are like 5,000 people that viewed my, I got two evolutionary biologists to make an entire 2-hour YouTube video about my 13-minute presentation.
Oh my.
And they got 4,000 people all hating on me.
No.
I gotta admit, on some level, I kind of love all the attention, even though it's negative. But all that to say, this is not a good place for a young lady to visit. But that being said, Reddit in general is just a cesspool. But there's certain benefit in just, it's at least a place I could meet people that want to argue with me. And that actually helps my thought process. And I get all this free editorial review. Oh my goodness, Katie. I can't tell you how many things that they've helped clean up and improve. And I don't even have to pay them.
Yeah.
So there is this creationist, Joe DeWeese. He got published, major peer review publication. And I'll tell you how that relates to what we just read. Anyway, this is the publication. It retails for $201 at Amazon, so it's discounted. And it's a book. It's like a magnum opus. It's a huge book. and it's by one of our own. And I am sincerely hoping, let's see if I can, that I can introduce you to him
Hmm.
because he may be working with us on this project.
Okay.
And the name Asharaf here. So even in this paper, that's one that's one of his fellow researchers, Asharaf.
Mm.
So that's a little bit of an aside. So there are things I might say that are a little mistaken, and he'll correct me. And uh, I'm going to just share this. So let's just do a little bit more and keep reading. This is also very helpful to me, but we'll start working on this. I want to start working from the same page, and then we're going to start using a little bit of BLAST. Okay, go ahead.
Okay. Mammalian topo-2 are inhibited by several classes of anti-cancer agents such as adrenomycin, amsaccharin, and
I have no idea what that is.
okay, etoposide. Hopefully, I
Epo-
pronounced those all right.
Etoposide is the way we normally pronounce it.
Oh, okay.
And
Etoposide.
Asharaf and Jodabis have worked with etoposide. Okay,
Okay.
let me just pause here just to explain. So, most likely, definitely etoposide is a poison, and it says here anti-cancer. So, if you take etoposide, you'll basically poison the topoisomerase, and then if topoisomerase doesn't function anymore, what do you think happens to the cell?
It probably dies.
Exactly. Unfortunately, when we have chemotherapies, about half the chemotherapies involve disruption of topoisomerases.
Mm.
So etoposide is one of the poisons. And by the way, the pharmaceutical company said, "Can you stop using the word poisons? This is supposed to be therapeutic."
Yeah.
So the chemist said, "Well, that's what it really does. It poisons the cell." And so there's a little bit of back and forth on, you know, softening the language, but the word is poison.
Mm-hmm.
Um, so anyway, go ahead. I'm going to go down so many rabbit trails if I allow myself. So well done.
Okay. Each of these inhibitors are known to interfere with the breakage or reunion of a reaction of topo-2 by trapping a covalent reaction intermediate, a reversible, cleavable enzyme drug DNA complex. However, some mutations and deletions in the C-terminal region of topo-2 of resistant cell lines result in aberrant localization of the protein in the cytoplasm. Nuclear import is selectively...
Go on.
Oh,
Go on.
okay, going. Okay. Nuclear import is selectively mediated by nuclear localization signal sequences and receptor proteins known as importance alpha and beta. The classical nuclear localization signal was the first such sequence to be identified and is characterized by a string of basic amino acid residues. An example is nuclear localization of the simian virus 40, large T antigen PKKKRKV. Bipartite, the nuclear localization signal comprises two strings of basic amino acid residues separated by a short intervening sequence. Like many nuclear proteins, topo-2 also has functional bipart nuclear localization signal motifs in their C-terminal
Oops.
regions. Okay, keep going.
Um. Okay. This is really good. Any? Why don't we move on a little bit,
Okay.
and then I'll invite some questions, or I'll just By the way, you have a very good speaking voice. Oh my goodness.
Thanks.
Um, I think you should occupy more of these shows because your voice is so much nicer to listen to than mine. Um, I kind of cringe when I hear myself. So this is actually, so basic, when they say basic, it's not like elementary. You have, can you guess what basic means there?
Maybe just like a short sequence. I'm not sure.
Okay, no, this is good. I think it means the pH level. Even though this
Oh.
is already an acid, acids have a certain pH. This is even more basic than,
Makes
so
sense.
that's what I think. And um, we can check that. But so I think um, so K is lysine and R is arginine. I think those are basic amino acids.
Mm-hmm.
Um, maybe we could do a quick Google.
Okay.
Let's do a quick Google. So I'm expecting to see arginine and lysine. K is for lysine. You know why they made it a K, even though it begins with an L? Because there's another one. There's another amino acid that begins with L. So they said, "Oh, we'll give it K." Just that for your information. So the
Okay.
list of basic amino acids is arginine, histidine, and lysine. So you'll start to learn the amino acid alphabet just by through constant use. So where did I put my? Oh
I
my
think you
goodness.
might have exited out of the article.
Okay, we can we can fix that. I'm so glad you have such good eyes for things. All right, so there's. I'm going to confess to you, and this is not at all unusual. Sometimes we're hoping we're interpreting a paper right, and that the parts we don't understand are not relevant to what we're trying to do. That's kind of risky, but sometimes that's what you have to do. So this part, this is this is all the laboratory stuff that they do, and I have no knowledge of that. So what they're trying to establish is they're showing how they established. So can you tell me what they were trying to do? I mean, just kind of you can kind of guess from the title. I mean, before they wrote the paper, what was their aim?
I think they're just trying to figure out what the nuclear localization signal is in this certain species of
Exactly.
fungus.
That's it. Perfect. This is what I like to. I mean, it's so clear what they're attempting to do, and so what they're trying to do to show the scientific world is their method of how they determined where the sequence was. And so this is just really detailed. They show all of their experimental protocols just to convince the people, hey, look, we did it. We went through all that we think we could possibly humanly do to prove that we really did find the sequence. And the magic sequence is, you can probably guess. Let me see if I can make this a little bigger. Positions 1, 2, 3, and 1, 2, 5, 6. From there to there. So now, so they looked all over the place. Again, this is almost not quite as bad as looking for a needle in a haystack, but it was still difficult. So this is the sequence.
Wow.
Okay, so let me try to explain this. So this is the amino acid sequence.
Mm-hmm.
And by the way, when it is first expressed, it almost always begins with an M, which is for methionine. Methionine. There's some alternative start amino acids, but it's usually methionine. Sometimes it can be deleted with some other processing. That's another story. But and the DNA The DNA, the codes for it is ATG. Do you offhand remember what A stands for?
Oh shoot. I'm
Okay,
blanking.
it's all right. All right, I'm gonna help you out here.
I know, like, what is it? Guanine, cytosine, threonine,
Okay, hang
alanine? Is
on.
it not alanine? It's not that.
That's all right. I have a cute thing I wanted to show you.
Okay. Wow.
Um,
I usually know it. That's kind of embarrassing for me.
it's all right. No, look, uh, I tell you what. I think I need to embarrass myself a lot more in front of you, and then you won't feel so out of place. Okay. So I actually don't have to try because I do that naturally. So let me try to. I'm going to share my screen and share my audio. I don't know how good the audio comes through. We'll try. These are my nephews, by the way.
Oh.
This is the DNA alphabet. A and T is for thymine. Now I know my ABCs. Next time he's in. This is the DNA alphabet. A is for adenine, G is for guanine, and T is for thymine. Now I know my ABCs.
Okay, I think I know it now.
Did you like that?
Yeah, that was cute.
Actually, I'm glad I actually did that because I get so confused with the very similar sounding chemicals. Because
Hmm.
A is for adenine, it sounds a lot like alanine, and I almost call that alanine. A is for adenine, T is for thymine, G is for guanine. So if you see ATG, that's the DNA. So you have the ATG here, and let me grab another genetic. See, and I actually have to refer to this a lot, and I think it's very easy to go crazy uh, looking at all this.
Hmm.
Oh, and that's not very clear. See if I can find a better one. See, they have a lot of them framed in terms of the RNA. Um, well, I think we'll just have to go with this one as best as we can. Let's see. I'm sorry it's so blurry.
No, it's okay.
So A, T, and G. Let's see. A, T, G. Methionine.
Hmm.
And then we saw here, get rid of this. The next one is serine. TCA. So let's look at that. Serine is T. That looks funny. It's here. It's supposed to be TCA.
It's under the C, like at the top. There's a TCA there too.
Oh, here.
Yeah.
Whoa! You have really good eyes. You're so quick. So there's a TCA here and then an AGT. TCA. So T, C, and A. Very good. See, there, I embarrassed myself. Now you're free to do the same. Um, I mean, we don't have to go through all this. I just wanted to show what this. Okay, this is why I say this could drive you crazy. Because can you imagine the poor researchers having to sift through all this?
Yeah.
And so what they had to look for was the sequence. They had to fish out. This was the nuclear localization sequence right here.
Hmm.
The reason I know that is I started yesterday Now, I don't know what these marks are. It sort of told me that's looking in the right place. So when we looked earlier, they gave the coordinates as 1, 2, 3, 4. So I actually had to go here. So I said, if that's 1,217, that's 1,218, 19, 20, all the way to 34. And then it ends at 1256. So, all right, Katie, do you have are you able to pull up a text pad, a notepad like this?
Yeah, I can. Yeah, I have a notepad.
Okay. Now, what I want you to do is I want you to lift off all of the characters in the localization sequence starting at position 1, 2, 3, 4. Now, since in the interest of time, I'm not going to force you to double check that I counted this correctly. So just type in from R all the way to K.
Like the codons or the proteins?
The proteins, the amino acids.
Okay.
There's a reason I'm having you do this because when we go to BLAST, you're gonna need to do this on your screen.
Okay.
Let me know when you're done.
I got it.
Okay. Now, do you have a dual screen system? Are you set up for 2 screens or just one on your computer?
I can try.
I mean, is there another screen set up on, a physical monitor connected to your computer?
No,
Right.
I just have my one monitor.
Okay. Okay, since we're recorded, if anything flashes on the screen that you want me not to show to the audience, I can edit it for you.
Okay.
So what I want you to do is show me your screen and get a browser up. We're going to visit the BLAST, a BLAST website, just like we did with Uniprop, we'll do BLAST.
Okay.
Okay, I'll try to do a share screen. Oh, it says present. So press present.
Okay.
And then press share screen.
Present.
You might have to play with it a little bit. Apologize for that. The reason I'm having you do this is I can be sure that you're able to, I could walk you through the website. It's going to, and how to actually interact with it. It's not like Uniprot where you're just clicking, pointing and clicking. Here you'll have to do a lot of data entry.
Oh, there it goes. Okay.
All right, I don't see it. Oh, wait, hang
Can you
on.
see
It's
it?
coming up.
Okay.
Alrighty.
Wow, that's a lot of screens. So I'm looking up this.
Okay, let me be sure I can. Let me see. Right now, I have to get out of. Okay, this is it. That's exactly it.
Okay.
Can you click on your man? You're fast. Oh my goodness, you're so much faster at this than I am. If you could show me what you typed into your notepad, let me see that.
Oh, I need to finish typing it out. I wrote it down on a sticky note, so I need to finish typing it out. Can you see
Okay.
that?
No, wait. If you wrote it on a sticky note, that's okay. We'll work with that. Okay, so go ahead and click on that. It says BLAST, Basic Local Alignment Search Tool.
Okay.
All right, so scroll down a little bit. So you can kind of guess what nucleotide blast does versus protein blast.
Yeah.
I mean, that's not that hard. So um, try protein, click on protein blast.
Okay.
All right, so we have this massive database of every protein we've sequenced or predicted. So we have sequencing a protein directly is immensely difficult. I mean, to take a protein, blow it apart, go through all the mass spec or whatever they call it, it's very, very difficult to get the sequence. So a lot of this is done with what they call protein prediction. So what they'll do is it's relatively easy compared to protein sequencing. DNA sequencing is easier. So they'll get the DNA and then they'll run it through some algorithm. It's actually DNA and the RNA, especially the RNA. And since you're familiar with the central dogma, you know how this plays out. The RNA has the messenger RNA, which can be translated, and that kind of you can then predict the protein, give or take a few things. So they have these massive gene sequences, and then they made a mostly predictive protein database of everything on the planet that people are willing to deposit in the database. So there's a lab out there. It's like, hey, it's kind of our obligation to society to let them know what we found. They'll just deposit it in this database. There are different levels of what they call curation. They'll have some that are considered, higher confidence and some that are low. And there'll be examples of that. I think we'll run into it when we do this. So when it says in that box, it says enter query sequence. What we're going to do is we're going to enter that sequence. It's going to search every organism and every protein that's in that database. It's just massive. It's insane. I do know of some DNA databases that have so much data. There's more data than there are galaxies in the universe. I don't know.
Wow.
Or at least someone has said something to that effect. I may have misheard him, but it's just gigantic. And that's just to preface that when you enter here and it searches, it could take a while. All right. So go ahead and enter what you put on your notepad.
Does it need to be uppercase?
You could put it in lowercase.
Okay. Does it need to be like, how's the spacing have to be?
It doesn't care.
It doesn't care. Okay.
So this is FASTA format, where we take the English alphabetic letters to represent the amino acids.
Mm-hmm. Okay.
All right. So now they have all these default settings, which I use once someone gets, starts to get very good at this. They may specialize.
Mm-hmm.
But all you have to do is just kind of scroll down and then just say blast. All right. So, all right, it'll say update in 2 seconds. That doesn't mean that it's going to finish in 2 seconds. It's just going to keep refreshing your screen.
Okay.
Now, when I ran, okay, so this isn't really running on your computer. It's running on theirs.
Okay.
So our hard-earned taxpayer dollars pays for the massive server farms that are doing this.
Mm-hmm.
So this is a very good project. I think it's beneficial to further into science. I'm very happy they made this available.
Oh.
Oh, hey, that computer likes you better than it does me. All right, so we'll get familiar with this. Even I have forgotten how some of this works because the interface has changed since I worked on it 8 years ago. Um, so, all right. So let me try to explain the columns.
Okay.
They always score. There's kind of, on a scale of 0 to infinity, how good was, how confident are we that this wasn't by accident, that we just stumbled on this. I mean, obviously, if you just entered the letter A, you could find lots of creatures and lots of protein sequences that have the letter A in it. It's not a big deal, but the longer your sequence and the more similar it is, the more confident you are that you're actually seeing a real pattern. Does that sort of make sense?
Yeah.
So now, one thing that I'm going to ask of you at the end is I might have done this even as recently as 4 years ago, but the interface has changed so much in the way, what it presents on the screen. It says show 100 and it only shows 100. It used to be I could just ask, unlimited. I think they started to say, well, that's kind of ridiculous because everyone's going to just be asking. It's going to tie up the servers for everyone. It's going to be a big traffic jam. There should be a way to tune this so you could get everything.
Okay.
So that being said, why don't we just scroll down a little bit and see what we have? Do you see humans in there anywhere?
Oh,
Homo sapiens?
I don't see any Homo sapiens.
Okay, so what does that tell you? The
That this
localization signal in Aspergillus is different than in humans. Now, what would happen if that localization signal is different for all the proteins in Aspergillus? Do you see what I'm saying? It has a different important.
Yeah.
And that important, if you change the important, you're going to have to change all like 300-400 localization signals and 300 or 400 proteins simultaneously. So going back, and I'm just kind of illustrating, it's like even if we assume common descent, do you realize, it's like I want to tell the evolutionists, do you realize all the things you have to change simultaneously? Not only will you have to change this localization signal, you'll have to change it for all the other proteins. And why would that happen? So this is where what the Darwinian process, I no longer call it natural selection. I try to avoid that term. Darwinian processes would actually prevent that because, a halfway working change is not going to be feasible. So like, let's say we just only changed half of the localizations and half of the nuclear proteins. That would be disastrous. And I'm just like, guys, don't you get it? So obviously they don't. So anyway, let me see. There is, can you scroll down slowly? Okay, some of these, look at uncharacterized protein asp we draft.
This one?
Yes, you have good eyes. Oh my goodness. So this protein has only 603. Topoisomerase usually has 1,500, maybe longer. So why does this one have 603? Remember I said people are just dumping what little they have. Sometimes you'll have an incomplete.
Yeah.
So let's see if you can go back. I hope the back key doesn't undo your beautiful search.
Oh, it looks like it's just in everything now. Well, let me. Oh, I went too far.
Okay. See if you can go forward.
There it is.
Okay, good. All right, this is going to drive you nuts because I didn't want us to have to wait. You know what I mean? To redo it over. I will say this: I am surprised about the length because the human topoisomerase 2 is only 1,500, so this one's a little longer. So let's just look at one of the. Just go ahead and click on that first one, like you have.
Okay.
And you see, that's 100% match. This is going to be, it's more interesting when you have deviations, but why don't we just go back a little bit? I mean, if you can page back, I'm sorry. See, this is what I don't like. I don't know, is there a way we can go back to where we were without, I don't know.
Yeah.
By the way, I wanted to say, do you see it says there, Aspergillus terreus, just above where you're highlighting?
Yeah.
See, that's the original one.
Okay.
And let's go down where it says, okay, we have some that have, all right, do you see that column that says like, I wish they had the title of the column. There's a column there that says percent identity. That really is a similarity.
Yeah.
So there's one like 95.65. Let's scroll down all the way down. So they're ranked in terms of similarity. So let's just go all the way down to the bottom. See if there's something that's really dissimilar. Oh, there's some that are 80. Oh, okay, that's interesting. Let's take one that's 86%.
This one?
Sure. And you can see that the score of confidence is a little lower, you know.
Okay.
All right, so the query sequence is the one you typed in. The subject is basically the protein, the record of the protein that's in the database. And so so your coordinates are 1 to 23. And then the coordinates of the protein, the Aspergillus homomorphous, it's 1302 to 1324. And then you could see that it puts an alphabetic letter wherever it's similar, but then it puts either a space or a sign, like a plus sign, where they're different. There is a meaning which I've since forgotten what plus means. It might be that like it's a similar kind of amino acid. It's not a radical change or something like that.
Okay.
I don't know where they put. I don't know how I figured that out. There must be like some sort of legend, but this is what sometimes having to learn how to use a tool entails. You just there are, what do they call it? There are free tutorials.
Okay.
Provided. So, do you have any impressions and questions or?
Not right now. It's pretty straightforward, I think.
This is a really cool tool. Can you imagine if we didn't have automation to search out these questions? Yeah. Yeah. I mean, this is glorious. I mean, the number of people involved to make that massive protein database, phenomenal. And then they make it available to us. It is so easy now to challenge evolutionary theory because we have these sort of tools and we can explore these sort of questions. So now can you press, all right, go up to your browser where it has the plus sign. I want to make another browsing window.
Okay.
By the way, it's great to see how versant you are. You're comfortable with browsers. Me and guys in my generation are just not as. Younger generation is so good at using computers. It's amazing. All right, go to the Uniprot website. I'm going to keep calling it Uniprot. I shouldn't be corrupting young minds and giving you mispronunciations, but I'm stuck in my ways. All right, let's look at see if you can find topoisomerase. Since you have gone through this once, why don't you try to download the topoisomerase 2 alpha for humans? Just try it. Okay, I'll help you out.
Okay.
Top 2A. Try top 2A. And we'd like humans. Do you think you can download or access the
this one?
Proteins? I don't know. Honestly, I think you'll get. You could try it, but why don't I save you a little bit here? Let's scroll all the way down.
Okay.
Because it might download what you're looking at, and I don't necessarily want to go there. It's been so they keep changing their interface, and the way I used to do things isn't the way it works anymore. So keep going down. This can you're gonna go go to the right, to the scroll bar on the extreme right, like almost off the screen. Move your mouse.
This one?
No, keep going.
Okay.
Right there you. Yeah, I think that's it. Keep going now. Go down.
Do I need to go to like one of these things on the side?
Not yet. Just apparently you're able to navigate this really well. Are you using the wheel?
The wheel? No.
On your mouse? I don't know.
Oh yeah. I'm using the wheel on my mouse. Yeah.
Very good. So just keep going down. Look at all those post-translational modifications. Oh my goodness.
Yeah, that's a lot.
That's very important to the function. It's just nuts. So keep going down real fast. Not real fast, a little faster, and
Okay.
where this is nuts. Look at all those post-translational modifications. Oh! Can you stop?
Yeah.
This is a teachable moment here while we're on post-translational modifications. Can you stop sharing your screen briefly?
Okay.
I just want to get on my soapbox a little bit here. Oh my, what happened to everything? Hang on. Okay, I got our photos back.
Okay.
How do you think a post-translational modification has to work? A molecular machine has to park in the right location.
Mm-hmm.
It has to find a needle in a haystack of all those letters. It's going to go through all around the cell. It's going to go into the nucleus. It's going to go look for a topoisomerase enzyme, and then it's going to find the right location. Can you imagine how difficult that is to navigate?
Yeah, I'm sure.
And then it has to recognize the way. I'm pretty sure the way this works is it has to recognize the sequence. It has to have some mechanism of recognition. Obviously, it has to recognize, hey, this is a topo. First, it has to be localized, right? The machine that will do if the post-translational modification is done in the nucleus. I don't know I'm pretty sure a lot of it can be done in the nucleus. Some of the post-translational modifications will happen in the cytoplasm, some in the nucleus, which is nuts. But the ones that modify in the nucleus have to get into the nucleus, so they need nuclear localizations too.
Mm-hmm.
But then they have to find the right sequence. they have to be able to recognize where in the topoisomerase it has to make a post-translational modification. So then you have another addressing problem. The amount of addressing and localization, or I guess all the transport processes in cellular navigation, it's just nuts. I mean, I don't know that anyone's totally characterized how even a fraction of this works. So off my soapbox. I mean, it just boggles the mind how complex all this is. So you can reshare. I'm sorry. I just got so inspired, as I see those post-translational modifications. So go ahead and share your screen. It takes a while, I guess, for StreamYard to bring it back up.
Yeah. Can you see it now?
Yes, fabulous. Okay, so let's just keep scrolling down. I'm sorry, I was just so moved to see how, when I saw that long list, I said, this is just, it's staggering. And this is just one of 20,000 proteins. Oh, you solved something. I can't do this on my computer.
Oh, I was wondering why it got stuck. Okay.
Oh my goodness. All right, so there's something about the way my computer's working. I can't see 3D structures.
You could probably like spin it. Maybe not.
Isn't that incredible?
Oh yeah, there it goes. That's pretty cool. Okay.
All right, you've actually solved a major problem. I didn't know whether it was the system or my browser or whatever. You're actually able to see three-dimensional structures. So that's called a ribbon diagram.
Yeah. Okay.
The longer name is Richardson ribbon, but that's incredible.
Yeah.
And oh, look at all that X-ray data. That, by the way, if you scroll back a little bit, just scroll back up.
Mm-hmm.
How do you suppose we get that 3D image? It's with the X-ray crystallography that helps.
Oh, okay.
And then we have computational predictions. So when they say PDB, that means protein data bank.
Mm-hmm.
And these are very difficult experiments to carry out because ordinary microscopes cannot see the enzyme. We have to use a lot of indirect methods, just for your information. So keep scrolling down. Getting close, getting more. Oh, there, stop.
This?
Right, so just scroll a little bit up. Okay, now the download will download what I know for sure I want to see. Those other invitations to download up there, all the,
Yeah.
I'm not sure this will give you what you need. Now, if you downloaded it, let's see, yes, the sequence.
Oh,
all
yeah.
right. So, can you cut or can you do a copy?
Mm-hmm.
You're so good at this. This is great. And then just do a copy, and then I'd like to open another browser. Okay,
Paste it.
type in BLAST.
Oh, put it in BLAST.
Yeah. But in your new browser window, go to your browser
Okay.
window, your new one. Yeah, it's probably not going to recognize that.
Yeah. I didn't think that through all the way.
All right.
It's a long
It's nice to see that you're eager.
Yeah. Okay, BLAST.
Now, honestly, I don't put it past Google to have actually found. If you put that in, it might actually find it, which would be funny. All right, so now I'm going to show you a little trick here.
Okay.
Scroll down a little bit. So now it says here, let's see. Oh, sorry. It says align two or more sequences. It's toward the top.
Uh-huh.
I shouldn't have made you scroll down.
Oh, I see it.
And you check that. Okay, so now you have two boxes.
Okay.
So you can paste what you just copied into the subject sequence.
Okay.
And I'm going to point out something when you go to the very top. You see where it says, okay, that little greater than sign that it begins with, that means it's a comment. So you can put anything there after after that, and it's just going to ignore it.
Okay.
So this is to help us. This is our identifier. So I mean, you could put your name Katie in there, and it's going to not care.
Okay.
Let's see. Although Katie. So in amino acids, I'm just sorry.
Am I?
A is lysine, A is for alanine, T is for threonine, I is for isoleucine, E is for glutamic acid. So you can actually do spell it in amino acids. Just thought I'd say that.
Yeah.
Sorry, I'm such a nerd. So now go to the other box up there, and you can guess what we're going to search for. We're going to search for the nuclear localization signal that was in Aspergillus and see if it's in human topoisomerase 2.
So you just type in like nuclear?
No. Type in what you had on your sticky pad.
Oh, okay. Give me 1 second. Oops. I lost my spot.
So you can see why it's good to try to use that regular notepad, because then you can keep reusing.
Yeah.
It's going to be a lot faster.
Okay, so just blast it.
Right. So we're going to test our hypothesis that a human doesn't have the same localization signals as Aspergillus.
Mm-hmm.
Actually, it already completed the search.
Yeah.
Because it only had to look at that. So now let's look at the percent. Let's look at the similarities here.
Okay.
If you can, yes, very good. Click on that.
Okay.
All right, for me personally, there is definitely differences. Now, that RK, I'm sorry, you don't even know where I'm looking. All right, the first one. So we'll go with the searches from beginning toward the bottom. So the top one, it says DDEDF.
Mm-hmm.
That's doubtful that is a localization signal. That's just random chance.
Yeah.
This one's a little more promising because it has RKRK, which is part of the localization signal. Remember the basic amino acids?
Mm-hmm.
Arginine, lysine, and there was one other one.
Was it H, maybe?
I don't remember.
I can't remember.
But this is definitely not the same localization signal.
No.
It has elements of it.
Mm-hmm.
So we want to see, anything with RK is going to be important. But so as a preliminary result, I'd say the localization signal is not the same. So let me, if you could stop sharing your screen. We're already off at 3:30. This was the preliminary. Let me try to get this um, get this back together. So the preliminary result is that the localization signals have changed between Aspergillus and humans, or whatever path, evolutionary path it took. Um, there's not, unfortunately, has been work that was done on topoisomerase, but I don't have the paper. I can't get it from my library. Normally, the library I get has good reach, but these are old papers that that are still printed, so I might not be able to access them electronically. So it would probably be worth contacting Joe DeWeese at some point to talk about this.
Mhm.
But we could always say, okay, we've outlined the differences. How could we resolve that there is a substantial difference enough that it would have, you know, uh, that a lot of changes have to happen to evolve from something like Aspergillus or whatever the evolutionists say is most similar,
Mhm.
on the evolution toward humans. Um, this is my thought. We could say, look, this is not definitive, but one way that we could get a more definitive answer is if we take the localization signal in Aspergillus and transplant it to where the localization signal is in topoisomerase. If it totally blows apart, I'm like, oh, okay, that confirms the hypothesis. It's not that easy to evolve.
Mhm.
Also, if we could take the important of a human versus a Aspergillus, that would even be better. Um, and those are the sort of experiments that are done in the lab. Um, Now, they're just, this is a bit of a sensitive subject. Uh, Understandably so, we can feel kind of uncomfortable that we're taking human genes and inserting them into other creatures. And I
Mhm.
totally get, if anyone's uncomfortable with that, with Christian background, I totally get that. Um, where I don't feel too bad about that is like the way we make insulin for, like my mom had diabetes, and the way they used to make insulin for diabetes patients, they would just harvest it from cows and pigs.
Mhm.
They'd take cow insulin and inject it in humans. When we learned how to do genetic engineering, we take the human gene, and the bacteria have absolutely no use for the human insulin. It's a signaling molecule. So the bacteria would make all this insulin or yeast, and then we'd harvest it.
Mhm.
Many creationists, myself included, think that's okay. I still feel a little uncomfortable, but that's okay. We can get around it. If you don't want to use humans, we could take rats, and
Mhm.
we've actually done that. We took rat topoisomerase and put it in a yeast. Okay,
Mhm.
so I just I just wanted to let you know. Uh, I am aware of ethical concerns, and if you have ethical concerns of anything that I suggest, please raise them up with me, okay? And we won't go down that route.
Okay.
But um, I do know a lot of creationists that will do stuff like this, and there are probably ways to do this at least a little more ethically. If we are just going around and changing the spelling, we don't necessarily have to transplant um, a rat or human DNA into an Aspergillus. We can, because we know how to genetic engineer. If we know the spelling, like you just know it, we could just make the spelling up with chemicals in our test tube, and then, so to speak, and then just put inside the Aspergillus.
Mhm.
So, you know, um, I'm just mentioning that, and I want to be sensitive, uh, to any ethical concerns, because we are, it does feel funny tampering with a creature's genome,
Yeah.
but uh, I did want to say is like this is the way they found that this was the nuclear localization signal was they didn't necessarily take the whole cell. They were able to take like parts of it and just uh, play to see if it would localize or not. And it's described in the paper. I'm not really familiar with the experimental protocols, but a testable prediction is there's a good chance that um, an important in an aspergillus will not work in a human, or let's say rat. Let's not use human, into a rat. Um, and we could always test whether a rat's important will work with an aspergillus. Um, that will at least tell us the degree of evolutionary change required if there has to be a lot of evolutionary change. Um, we could see if, like, say, 400 of the protein, 50% of the proteins that should be localized aren't being localized anymore when we make that change. I think that'd be an interesting thing to pursue as as research. But so maybe for next week, if you could okay, this is where I really don't have any guidance for you except I'm going to rely on you to do this.
Okay.
You can find a class that will teach you how to do BLAST. Like they have like maybe 5-10 minute tutorials somewhere, and the government actually makes some of them. Then there are people who make them independently, privately, that you could find on YouTube. I want to know how we can find more than 100. Remember, we only got 100 hits in all aspergillus. I want to find out how we can make that list bigger. I literally was able to reset the parameters once. I was able to get 5,000. I think up to 20,000. I mean, the machine cranked away for a long time. I think it took an hour to get 5,000. But I was writing a paper related to that, related to a project that needed that. I wanted to survey 20,000, just so I could report this is how many hits we got for this. So if you can figure out how to do that, you could also show me.
Okay.
And I may or may not send you a paper that I read on nuclear localization signals, because you mentioned one particular word that you didn't understand, and that was bipartite. And there was a paper that explained it, but maybe we don't need to necessarily go there. But anyway, do you have any thoughts you'd like to share before we go backstage?
No, I don't think I have any thoughts today.
Okay, so we might try to just look around for, it depends on how successful we are with BLAST. And whether we could find coordinates of localization signals for other creatures, Is there any creature that you're particularly fascinated by?
Well, at school I do research on fruit flies, so.
Okay, so there we go. Oh, that's great. Okay, most of mine has been humans because that's where all the interest is, and so there's just more data. So we'll probably survey eukaryotic. Well, actually, prokaryotes don't have localization signals, so that'll make it easy. So you can try to survey literature as you have time, and I know you're busy. Localization signals of other creatures. Now, sometimes, I mean, it may have been reported on Uniprot somewhere. If you're looking at one particular protein, I don't remember. Now, for what it's worth, we could spend weeks looking all the topoisomerases out there on the planet and Actually, we could spend more than several lifetimes if we wanted to look at all the topoisomerases of all the creatures that we catalog. That would be impossible, virtually impossible. Um, we can use the computational tools to help us, but it at this stage of even my understanding on how to do research, sometimes you just have to go to the papers like what we did.
Mm-hmm.
It's not quite as there's. I mean, if you Google and you find a database of localization signals, that would be great. So could you put that on kind of just to find out if there's any database of localization signals? If not, you and I are basically building a small database of localization signals. I mean, it doesn't take long to start to be on the cutting edge now that we have computational tools. Cutting edge means we might be asking questions that no one else cares about. um, and that would be great in and of itself, because if you report that there's not any database of all the localization signals, we're contributing. We're now starting to form a small little collection, and I think that would be beneficial in and of itself, because we have all these creatures and we have all these proteins, and um, you know, there's basically not any limit to what we could explore uh, with tedious research like this. So um, I think this is a good framework. It's flexible. You can research as much as you want, um, or as much as you're willing to take. I mean, um, I uh, I will tell you, I've seen some of these studies, and it's like these guys had the patience of Job, because they would go through so many creatures to just do all these comparisons, and I'm like, that would kill me. Even with automated tools, they probably had to make automated tools.
Mm-hmm.
Wow. So anyway, I'll give you the last word before we go backstage.
I don't know. I guess thanks everyone for watching then.
All right. Take care, everyone. God bless.